It's my pleasure to introduce this evening's program, Epigenetics Changes Everything, Revisiting the Nature versus Nurture Debate, and our speaker, Dr. David Allis. A distinguished biochemist, David is Rockefeller's Joy and Jack Fishman Professor and head of our laboratory of chromatin biology and epigenetics. He's made essential discoveries about molecular mechanisms that govern the activity of our genes, which will advance our understanding of conditions ranging from birth defects to cancer. David is a member of the National Academy of Sciences and has received numerous distinguished awards, including the Gardner Foundation International Award, and just this past year, the 2015 Breakthrough Prize in Life Sciences. After David's presentation, Alan Alda will join him on stage for a discussion and to take questions from the audience. Alan is, of course, known to most of us for his iconic role as Hawkeye on the long-running television series MASH, and he's also famous for his work on the big screen, such as his Oscar-nominated performance in The Aviator. What some of you might not know is that Alan is also a master of communicating about science. For more than a decade, he's hosted Scientific American Frontiers on PBS, and he's brought legendary scientists to life on stage through his portrayal of Richard Feynman and his play about Marie Curie. He's also played an active role in the World Science Festival as a board member and past co-chair. In 2009, the Alan Alda Center for Communicating Science was established in his honor at SUNY Stony Brook's School of Journalism. As a member of that group, Alan's been active in encouraging scientists to be more effective in communicating their own stories of discovery. Tonight, we're delighted to bring David Allis and Alan Alda center stage at Rockefeller to tell the story of the epigenetic discoveries that promise to transform biomedicine. Please join me in welcoming Dr. David Allis.
Am I coming through? Hopefully loud and clear. This is an evening of communication, so if I can't even say hello, we're in trouble. It's my honor, absolutely, to be here, and rather than single any of our many distinguished guests out, because I'll forget someone, I'd like to welcome newcomers, seasoned other past Rockefeller associates, and all the very distinguished guests in the audience. It is my pleasure to give this talk on some of the aspects of what my lab is all about. So tonight we're going to talk about this sort of unusual concept because all of us, I think most of us, took enough science to be completely convinced that genetics matters. And since this is about celebrating science with parents as well, we think about what we inherited from our moms and our dads and so on and so forth. I hope I can convince you there's some new wrinkles and things that we need to think about. So this is the oldest cartoon I could find. This even predates my birth, but I have to say not by much. But it is, I think, a situation that at least I know I can relate to, standing in front of my parents and with my report card being scrutinized where the child says, I assume to his dad, what do you think the trouble is with me, Dad? Is it my heredity, which I'll transfer to, say, the genes, or is it our environment? And that'll be sort of the epigenetic part of my talk. So I think I want to start with a breakthrough discovery at Rockefeller. A little bit of history, but it's an important discovery because this was game-changing. We all are used to thinking about genes, but it was this group of scientists right here at the Rockefeller Institute, it was called at the time, from the hospital on campus, where Drs. Avery, McLeod, and McCarty all discovered for the first time that genes, which we think about and are depicted here in this blue box, genes that make us, if you will, who we are made of DNA. In this point in time, around 1944, it was contentious what was really the transforming or the heredity material, and they proved unequivocally that that substance is DNA. This predated the Watson and Crick discovery of the double helix's structure. So absolutely, I want you to think about real genetics. As you well know, and I'll say in a minute, the genome has been sequenced from us with the idea of understanding who we are. And it's a little bit complicated, but it was very clear that there are regulatory sequences in DNA, And there are various factors. We'll just keep it simple. Protein factors that engage the DNA template and turn genes on or off. Here I've said activators. Turn the gene on. And what I want you to note is that the DNA, the gene, is relatively fixed. We can't change much about what we inherit at the level of DNA. So hold that thought. In 2001, roughly 60 years later, from the Avery-McLeod-McCarty discovery, our genome was sequenced, and this was truly a tour de force. I'm sure you all heard about it. It's now been over a decade, and here was the promise. It was suggested to us that by knowing our genes, our entire genetic makeup, our blueprint of life, genes determine disease. That's a good thing. Genes determine aging. I really need that to be solved. And importantly, genetic analyses were going to give you hints into things that would provide potentially insights into potential diseases and therefore potential therapies. Now something that's been is happening quite regularly, especially here in this neighborhood, personalized medicine, or now it's been called precision medicine. But it all was centered on understanding our genome. So what did the Human Genome Project not deliver on? Several questions. One, there was only about 21,000 genes. That may not mean a thing to you, protein coding, I should say, genes, but that may not mean anything to you, except that's about the same number of genes that exist in worms and flies and fish. And that doesn't seem right. That just doesn't seem right. So, I'm not saying we're at the top, but nonetheless, where does individual variation come from? We're all different, but what about monozygotic twins who are genetically identical twins, and yet one has potentially a completely normal existence, and one individual, the sister or brother of the identical twin set, has got some disease? What gives? How can past experience in our environment, back to the cartoon, shape who we are? And importantly, perhaps, on the theme of making diseases go away and make us better, are there some diseases that don't have their foundation in mistakes in DNA? So this is going to lead us to tonight's centerpiece topic. Epigenetics is a reasonably old word. It's the combination of epi, the Greek prefix, which means something over, above, or in addition to genetics. And at least for starters, you might think about it this way. If you think about the English language and you're willing to equate a word, an individual English word, as potentially a gene in our DNA, then you might sort of think of epigenes as how those words are read. And without changing the letters, you might make a bold-faced word. You might italicize a word. You might underline a word. You don't change the letters, but you would change how the words are read. For those of you that are more tech-savvy, genes might be thought of as a hardware computer, epigenes as the software. And that's how it's now thought of, hard genetics versus soft genetics. But this cover of a journal article not too long ago was dedicated to the topic of epigenetics. And the artist that put this together, I think, is trying to say how many vast and very interesting aspects of biology seem to have this non-genetic aspect to it. And I'll talk on really only two today. That doesn't mean the other puzzle pieces aren't interesting, but I'll make all my comments are centered around disease that might chase back to epigenetics and environment. So that's going to kind of be the nature-nurture aspect of it. Now this analogy is not meant to offend anyone because it's so simple-minded, but it works for me. If you baked a cake, that's got a particular taste. But what if you add different icings? Again, something above the genome. Now you're talking about actually above the cake. And that will generate different tastes. You didn't change the cake, you changed the frosting. In biology, it's always been fascinating to myself and developmental biologists who coined the word epigenetics, that once sperm meets egg and you have a young embryo, that embryo, with all of its cells, has an identical genome in every cell. But over time in development, that embryo is going to give rise to potentially a vast array of different cell types: blood, nerve, bone, skin. And of course, some of you know the raging interest in stem cells that are kept in the body almost in reserve, who are somewhat able to do this differentiation again. But it all comes from one genome. So what else might be going on if the genes in all these cells of all these cell types is the same? And of course, unfortunately, mistakes made in this epigenome layer can take the cell backwards to even a more undifferentiated state, causing that cell to enter a proliferative crisis, leading to a cancer of some type. Now, I should have thanked you all for coming on a weeknight. It's a beautiful night. But to just keep everybody awake, this is a cartoon from one of my former postdocs, Sean Taverna. He's now an associate professor at Hopkins. Sean's hobby was drawing cartoons that were sort of science-oriented. I love this one. This was the first he ever gave me. And with no offense to the women in the audience, the idea here is that DNA steps out of the shower. It's got to package itself. It's got to wrap itself up. And it's going to make a choice for one of two towels: histones or her stones. And it just happens that what my lab studies happens to be the histones, but I would have been okay if it was a her stone as well. So what are histones and what do they do? Well, a lot of you know our genetic material resides in the nucleus, and if you were to pull out a long spaghetti fiber, DNA, the Watson-Crick double helix, the discovery made here at Rockefeller that genes are made of DNA, it would never fit inside the nucleus. It's about 2 meters in length, and clearly the cells of our body are on the order of micrometers. But to package this DNA, nature evolved a spool around which the DNA wraps. You can see 2 turns, and that, if you will, bead on a string, we'll think of it like a pearl necklace. It goes to another unit, and then it starts to wrap further and further. That structure of histone proteins, that would be the balls, if you will, around which the DNA wraps, the DNA, that complex is termed chromatin. And that's what my lab studies. Now, it wouldn't take a rocket scientist to think about how hard it would be for a gene to be expressed. How would the proteins know where to go? How would they know where to activate or where to silence? So you might envision that this kind of more open chromatin, where you might think of it like a slinky toy and you've pulled the slinky apart, it would be far easier for genes to be expressed here. So these genes might be on. In contrast, genes that were in this highly compacted state depicted here might be really compressed, compacted, and shut off. And I'll show you in a minute a close-up of this repeating unit, but the question from my laboratory was, are there molecular switches that would throw this switch and enable this chromatin template, which is our physiologically relevant template. It's not naked DNA like the cartoon. Wrapped with these histones, the tau, how would you molecularly open and close that fiber? So here's that repeating unit. Again, just to repeat, the histone proteins are inside, making the spool. There's 4 different types. That's why there's 4 different colors. That doesn't matter. But I hope you can see a little bit of the DNA wrapping around the outside. And what was striking is these histone proteins have long tails. And it doesn't take, again, I think a rocket scientist to think maybe that's where the molecular switches lie. Because the cell could throw them on or off by putting something, a flag or a tag, on these tails. And that's my second Rockefeller discovery. Vince Alfrey, shown here, in 1964, so just last year, the scientific community celebrated the 50th year of Vince Alfrey's discovery that there are chemical tags or flags, off or on, but I'll refer to these in my talk as modifications, that are literally attached as signals on these histone tails. So again, take a breather. Histone proteins are proteins. They themselves have to be encoded in DNA, but they serve to wrap the DNA, and here's these potential molecular switches. And I think it is my lab's probably best known discovery, if you will. I'm not the brag type, but in 1996, my colleagues and I were fortunate to access for the first time the enzymes, the proteins, who are responsible for either adding these marks, so I like to call them writers, or in contrast, enzymes that oppose this activity that strip these marks off, and again, we might call these erasers. And as Alfrey had hoped and dreamed, these were the molecular switches. And the flags that he discovered here at Rockefeller, off or on, although he didn't know it, these were the switches that turned genes on or off. And after the community followed up on who these writers and erasers were, many of them, most of them, were dysfunctional in human cancers. And that began to change everybody's thinking about not just DNA. So let's take a sort of this is the take-home sort of summary, catch your breath. So we started by talking about real genetics. I sort of lied and said the genetic material is fixed. Not quite true. If you too much sunlight, too much cigarette smoking, you can pick up mutations in the DNA. And of course, that's a bad thing. But those mutations are hard to fix. Once they're in the DNA, you will pass them on to your children. That is genetic heredity at work. And so everyone, when I was sort of a postdoc, everybody and their uncle at medical schools wanted to try to do gene replacement therapy. How could we fix these mutations? But if you contrast what's happening on the epigenetic side, if there's a mistake made with these flags, with these tags, you've never changed the DNA. The DNA is 100% as it should be. And so there's a promise that maybe epigenetic mistakes would be reversible. And a big inspiration for me to move to Rockefeller was to be not just here at Rockefeller, which is amazing, but you saw from the science fair, you hope these young kids become potentially somebody in your lab. But across the street at Memorial Sloan Kettering, epigenetic drugs were developed. They were put in humans. And this is the first FDA-approved drug for the first ever epigenetic therapy in humans. This is a gentleman at Sloan Kettering who was treated with a cancer in his larynx. The larynx spread to his lungs. This is the individual, 2 different chest cavity scans with large tumor masses. At this scan, a very large tumor mass here. This is before taking the drug. 8 weeks later of just an oral administration, you can see these tumors have greatly sort of recessed and become sort of dissolved away. And now there are 4 epigenetic drugs that have been FDA approved that are helping to help patients live healthier lives, again on the promise that mistakes made in this tag, this flag system, are reversible. Now this is fairly current. This is now, for parents in science, this is a moving admission that I'll make to you that I have grandchildren. My wife's an elementary school teacher. She's here. But at the close of 2012, 2 laboratories, not my own, found game-changing mutations. So let's stop. That's DNA mistakes identified in pediatric cancers. So that makes it a childhood problem. These 2 discoveries were made by Susie Baker and her colleagues at St. Jude's Children's in Memphis, Tennessee, and Nada Jabato and her colleagues at McGill University in Quebec, Canada. And right away, we were absolutely, why do science? We were here at Rockefeller, and we read these papers in December 2012, and I knew right away we had a calling, so to speak, because these childhood cancers are brain cancers. Worse than that, they're cancers of the brain stem. For the experts, it's called DIPG. For diffuse intrinsic pons, that's the brainstem glioma. And here's the tragedy. Once this diagnosis is made, and which children does it fall in? On average, usually 5 to 8-year-old children. These children are presently universally fatal with a survival time of roughly a year or 2 tops. And the mutations that these Baker and Geppetto labs uncovered was in the histone protein. Of course, if you're following me, it's in the DNA that encodes the histone protein, but nonetheless in the histone protein. And so the severity of this disease has inspired my laboratory and now many worldwide. There's no treatment for these children. It's a death sentence. And here again is that fundamental repeating unit of our genome, packaged histones with DNA. And here's these sort of histone crazy tail domains with these modifications attached that work to flag on and off signals that Alfred first turned us on to at Rockefeller. And I honestly wish this was a test question. Where do the mutations map in the histone proteins? I kid you not, they map right to the amino acid that attaches the signal. For me, it doesn't get, I don't want to say better, it just doesn't get more important than our lab's ability now to swing into action. We've termed these mutations as oncohistones. The frequency of these mutations in the genome is so high, it's now believed that these actually cause the cancer and drive the cancer in these children. So what have we done about it? Well, I go give seminars. I'm blessed to give seminars. And in the December of that year, I traveled to Stanford, California to give a regular Allis seminar. But there I met for the first time Michelle Manji. She's a physician scientist at Stanford Medical School. And in my office chat with her, she knew about now these oncohistone mutations. She wasn't a chromatin or a histone lady, but nonetheless, she said, I've got a patient. Now, I wouldn't feel good showing you this, but this family, the Krantzes from San Francisco, and their 6-year-old daughter, Jennifer, were under Michelle's care. And Michelle told me that Jennifer was probably going to live only 2 more weeks. This family made a documentary about their daughter and her struggle and her final passing. And it was their hope that this would actually stimulate funding and research on the DIPG cancers, the brainstem cancers. So sadly, Jennifer did pass away 2 weeks after my visit. Michelle Manji wrote me and said, I can't keep doing these brain tumor harvests, but here's the tumor. And what did we do with it? The top is the control. If Michelle sent us brain tumor samples from children who did not have the genetics of the DIPG histone mutation, they stained very, very well with, this is too much detail, but with an antibody that we raised in my laboratory against the modification. I hope you can see lots of brown nuclei. Jennifer's tumor sample, in contrast, and others like it, when stained with the same antibody, they just don't stain with this modification. They just don't stain. There's no exception to how black and white this is. My laboratory, as Mark mentioned, is a biochemistry lab. Using these tissue tumor samples, I'll just give you the take-home, made the remarkable discovery in 2013 that this mutation in the histone tail is literally causing this result because it's completely inhibiting the writer, the very writer that puts on the mark. No wonder there's no staining here. But this gives the community at large, for DIPG scientists and persons like myself, an insight. Now we have a therapeutic target to go after. We need to override this inhibition to get samples in, hopefully, mice and then children back to the normal. And now, to bring you up to date, as of this minute in time, it is no joke, it is truthful, that pediatric cancers of the blood, the brain, and the bone, all different cancers, but interestingly, tragically, all children, each have a mutation at a different position. So the blood cancers are showing a mutation at the extreme end of the histone tail. I already have talked to you about the original brain cancer mutations here in the middle, and the bone cancers are mapping to a position in the base of the tail. All amazing to me. All of them we've gone on to show, not even published yet, they all inhibit their writer. their own writer. So we have potentially not just a tragic, tragic brain cancer that we're keying in on, but some bone cancers and some blood cancers as well. And although she's an outlier, she's an inspiration. If you follow sports, or maybe just national, international news, you might have heard about Lauryn Hill from Cincinnati, Ohio. I think she was the oldest DIPG patient, but once she was diagnosed at 18, she went to her college, and more than anything, she wanted to play one basketball game, potentially before she passed. And that's exactly what she did. She raised a tremendous amount of money in a contest, a challenge, kind of like the ice bucket challenge. Lauren's challenge was spin around 4 times at the free throw line, get as dizzy as I am all the time, and try to shoot a basket. And whether you make it or not doesn't matter. Then get somebody else to do it, and so on and so forth. And she raised over $1 million for the fund, The Cure Starts Now. All of that is based in Cincinnati, Ohio. She did pass away from this disease in 2014. Wheaties put her on the box. She truly, in my mind, is a champion for sure. Her motto was never give up. And I'll just tell you as a footnote, anecdotally, I was born and raised in Cincinnati, Ohio myself. So this is really quite remarkable for me. You might think this is only cancer. It's now exploding into a large number of different non-cancer diseases. Don't worry about the papers, just worry about the headings. Single gene diseases, neurology, fibrosis, cardiology, immunology. There seems to be many, many epigenetic links where you haven't changed the DNA, you're either misactivating or you're missilencing the genes based on mistakes in the epigenetic landscape. And Watson himself said, at this point in time, you can inherit something beyond the DNA sequence. That's where the excitement is now. At the same time, Our Time magazine put epigenetics on the cover, and their centerpiece article shown here, Why DNA is Not Your Destiny, is what I'm going to talk about at the end of from now on, which gets to the author, John Cloud, saying the science of epigenetics reveals how the environment, so now, the environment, this would be the nurture part, and the choices you make change your genes, but unbelievably, those are your children. So the lead article features this family from Sweden. It looks genetic to you, right? It's the grandfather, father, and son. But the star of the article is the grandfather, who's a pediatrician from Stockholm, Sweden, Lars Bygren. And it was his careful studies of families in the most northern part of Sweden that occasionally underwent feast and famine issues, where he'd be able to find out that not only were the parents and children affected in ways that meant weight, lifespan, disease susceptibility, mental health, but none of this tracked their genetics. It tracked their diet, and it tracked precisely whether it was a time of feast or a time of feast or famine. And I have to say that his very careful record keeping pointed a finger as the dad's the troublemaker. And I think that's why they photographed the family this way. So last year, a landmark paper, not from my lab, from a German group, did this kind of experiment in the fruit fly. Made the cover of Cell, a prestigious journal in our field, just feeding dads or moms 2 days worth of high sugar diets caused the children of the next generation to be obese. And here again, it wasn't the moms that seemed to transmit this problem, it was the fathers. So their molecular analyses, which I'll spare you, took them right to marking key genes that control weight with mistakes made in this epigenetic marking system. I wouldn't feel fair to my male colleagues in the audience, perhaps. The problem isn't just with the fathers. When others took pregnant mice, all genetically identical, and simply fed the mothers when they were pregnant different diets, that contain not, in this case, not sugar, but different modification enriched diets that are the modifications that these histones get marked by. Their children, all genetically identical, changed weight and clearly coat color. So this was a problem that was somehow happening to the young fetus through the pregnancy, through the mother's line. And Michael Meany, who was trained here at Rockefeller with Bruce McEwen, has gone on at McGill University to ask kind of, I think, a very far-reaching question that a lot of us think about from sort of the news. If you're a good mom, does it help your children? If you're a bad mom, perhaps, does that hurt your children? And the Meany lab has set up mice that are either extra good mice, meaning they lick, they groom, they nurture, they care, or bad moms. I won't say what they do, but they don't behave well. And these crosses have now shown the Meany lab and others that whether you're a good or a bad mom, so parental care is also impacting the epigenetic marks that are then inherited and transmitted, affecting brain factors that in turn affect stress response. And you can imagine what the human ramifications of this are with respect to good moms versus bad moms in us. So this is my almost final slide. It's my big wrap-up slide. So I don't mean to dismiss genetics. Please understand that. Genes are critically important. They do make up our genome. They are the basis of true genetics. And except for mutations and chromosome breaks, they don't often change. But should they change, it's hard to rectify them. It's hard to reverse them. Sunlight, smoking, no doubt, not good for us. But there's a curse and a blessing to the epigenetic layer. These covalent, these chemical marks, whether they're added to the histone proteins or they're added to DNA itself, they form together the epigenome. And it's remarkable that they're not in DNA. They are inherited. And the good news is if mistakes made here are bad for us, there's a prayer we can reverse them. So that's a promise of, say, cancer therapeutics or cancer epigenetic therapies. But the bad news is if we eat things we would have said, well, that's just our problem. We were bad to ourselves, pay for your own sins. But now it's turning out that may impact your children or your grandchildren. And for me, that's kind of a sorry, kids. So this is my final, the overall, this is for someone very special. We're going to segue to his role tonight. The big take-home, the serious take-home is modifications of histones or DNA that I've been talking quite about do alter the science of heredity. I think both nature and nurture are playing roles in us. And of course, you'll all pick up on the fact that's just MASH in disguise. I have lots of people to thank. We have much support from various institutions, but I know some of the people who have really been so generous with their vision and their foundations are in the audience, the Robertson Therapeutic Fund and so on and so forth. And of course, we get national funding from the Cancer Institute, and none of this would ever have happened, especially when we were between grants or whatnot, without very generous support from Rockefeller University. I'm grateful for Mark for giving me the chance to represent the school, and of course, the development office, the entire folks that were responsible for tonight. And thanks to them and thanks to all of you, for sure. On a weeknight, give me a break. Thank you very much.
What a wonderful education in just a few minutes. I'm interested, because it's fresh in my mind, about the M and the A of the MASH acronym.
Oh.
Thanks for the acronym, by the way. No. You can't miss with that acronym. The M, the modification, and how it alters heredity. The DNA, little bits of DNA wrap around these histones, proteins.
Actually, quite a long strand of DNA.
A long strand. It goes around many times.
It'll go from one unit of that sort of bead on a string to a second bead on a string to a third and so on.
Right.
And that will all compress.
So the modification that takes place is something that happens to the histone, right?
Absolutely correct. It's,
And
it's a
especially-
chemical flag that I spared you. I just called it mod.
Yeah.
Alfred taught us what it really was in terms of real chemistry. It can have on flavor or off flavor, but it goes on the histone protein.
And the reason that that little flag, the tail comes out with flags on it, right? Am I right so far?
The tail is just part of the histone.
Right. And the reason it sticks out like that is so it can do something.
I think so. I think so. And,
One of
you know,
us better know here.
Well, and so, you know, in the laboratory, well, I should have said, let me just say something quick. Histone proteins, those proteins that package the DNA, in all organisms that have a nucleus,
Yeah.
it is the same from yeast to man. Nature hasn't changed those histone proteins at all. And even those tails that you saw were not closely associated with the DNA, they were protruding out, they are absolutely conserved. So if you say, well, but wait a minute, they should be dispensable. I didn't see them really wrapping the DNA. The DNA was tighter. No. Nature hasn't changed those tails at all.
So does that tail on each histone come in contact with the tail on another histone? Is that
That's
how
a
it
great
does its
question.
work?
Yes, absolutely. The tails have been floppy, so it's been hard to see them in structural analyses. But in the few cases where they've been able to track where the tail goes, you're spot on correct. It does go to an adjacent, if you will, spool, and it will serve to help compact.
Now, how does it go anywhere? It's all tied up with the surrounding DNA that's
Well,
wrapped
that's-
around its own histones, and they're all lumped together. Where does it go?
Well, it seems to be able to at least flop or bind or compress or compact up against other regions in the genome. And then these modifications, you can think of it just almost like unzipping a zipper.
Right.
I mean, it's too much detail perhaps, but DNA as a molecule is very negatively charged. These histones are very positively charged. And these tails are super positively charged. And the alpha modifications, they attach and negate some of that charge. So I think it was Alfred's dream that these might explode the chromatin to open, or perhaps when they removed the marks, the chromatin would compact back.
So a modification takes place, and it doesn't signal the way it's supposed to. Because of wrong.
There are mistakes made.
How genetic modifications take place when there's something goes wrong with the DNA.
Correct.
But here you
Absolutely
have
correct.
something going wrong with this little protein. What's the difference? You talked about sugar can affect a couple of days of high sugar diet can affect the histones.
Through these modifications.
But sugar also affects the DNA sometimes, doesn't it?
It absolutely can. But I think
So
in this,
what's the difference? Why doesn't it go for the DNA? Why does it go to the histone?
in fact, it might. In fact, it might. I think that these authors in that study with the flies having the high sugar diets, they actually track, because flies are very good for genetics, they actually then asked, well, what mutations would disrupt that obesity inheritance? And they tracked it right in on those writer proteins that add these chemical marks. So for me, once a laboratory does a definitive genetic experiment, you then absolutely become sort of more of a believer that at least the histones are at play. It doesn't mean the DNA is not at play.
Right. But here's what the question really raised itself in my mind. You talk about these things being heritable, the mutations in the histones. But what really interests me is if I see this really interesting pie and eat the whole damn thing, have I just destroyed my grandchildren's lives? I mean, how long does this last?
Frankly, I doubt it. But in the laboratory where it can be studied in these models, and even the physician who very carefully did sort of the careful background work in Sweden, it looks like it goes on for about 2 or 3 generations, and then I don't know if it peters out, but we know enough now about these epigenetic regulators that they do have a certain sort of finite amount, and they will eventually get diluted out as cells replicate and replicate and replicate.
So they lose their power over time.
Lose their oomph, right,
Lose
over
their
time.
oomph. Does that mean that in order to create a modification that's strong enough to be heritable for 1 or 2 generations, you need more than one pie?
Yeah, you might think of it that way. So eat away.
Yeah, so, I mean,
But in all seriousness, in the fly organism, which is so powerful, these same experiments, you can now not just make a mutation or a deletion, remove something, you can actually overexpress. And that will cause the inheritance to go longer. So all of this, I think, does point a finger at some level for mistakes, up or down, on these marks can influences that are coming in from diet, exercise, other potentially.
So it sounds like when you bring up exercise, I can see how maybe the absence of exercise plus a bad diet could have a deleterious effect.
For sure.
If you add exercise and have a lot of exercise and good diet, can you do a favor to the next generation?
Well, that's a good question. I think Bruce McEwen, who might be here, might actually have knowledge about that. But I think the Meany Lab, who I sort of wrapped up my talk on with the grooming or good or bad moms, I think those kinds of experiments are fascinating and they're ongoing. So, I know Bruce has absolutely given lectures here, probably at the Parents and Science many times, but also to the Rockefeller community. And, exercise is a good thing for sure. Whether and how it might impact and work through the epigenome, I think is still an active area of investigation.
I was very interested to see your mention of the first epigenetic drug. That's news to me. I didn't know there was anything like that.
That was developed, again, right here at Sloan Kettering. Paul Marks was the president at the time at Memorial Sloan Kettering, and it was his group that really championed, people at that point in time were worried. Think about it. If you start to throw a drug on something that's, this fundamental packaging system in our genome, the naysayers were, oh, you're going to cause everything to be deregulated. This is going to be toxic.
How did they know they weren't going to do that?
I think they didn't. But he was smart, and his colleagues, and he stayed the course and developed cellular models, then moved it into mouse, got some intriguing data that this just wasn't willy-nilly, misactivation, and everything was haywire, and then convinced some small clinical trials to happen, and bingo, some of these cancer patients improved right away. And so
is the process to examine it and find out how the histone differs, how the code differs in the histone, and see maybe there's one letter missing, and you stick that letter back in, or how do you do it? How do you fix it?
Well, I think the pediatric tumors that I showcased from the very tragic DIPG patients, that was post-death, autopsied sample material, because it's so hard to get these biopsies. It's just dreadfully awful. And that material was sequenced as a genome. And of course, in the personalized medicine, the precision medicine, you'll go through and you'll find mutations, and they found them in the histones. Now, most people would just dismiss those. Histones, come on, give me a break. Histones just package the DNA. No, they stayed on and got more and more data, more and more children, tragically, and the frequency of these mutations in the histone genes were unbelievably high. In some of these patients, it's a 70% frequent mutation. In the bone cancer, pediatric bone cancer, it's now pushing 100%. You can't find this bone cancer without the histone mutation.
So this may be too detailed, but I'm really curious. What do you do to remedy it?
So, I'll just say as a modest, very modest progress report, I sort of said as a punchline, these oncohistone mutations turn off and inhibit the writer. So the writer, again, is the enzyme, the protein enzyme that has the job of putting on the mark. So the writer.
Tell me something basic. What's the role of an enzyme compared to a protein?
An enzyme is a protein.
Yeah.
And all enzymes, for the most part, there are catalytic RNA molecules that made some.
So the proteins that do
But
certain
in general,
jobs.
proteins do many, many jobs, some of which is to facilitate a chemical reaction. You know, and in this case, the enzymes that we're talking about hold on to the modification. Again, that's a chemical group. They will learn to engage the histone tail, that's their substrate, and they'll transfer the chemical group onto the tail and leave. So that would be a writer. An eraser would come along and say, oh man, I got to remove this and take it off. So those are proteins, but there are many, many other jobs that proteins do. But enzymes, for the most part, are protein-based.
So you can send enzymes in, and they'll, if they're the right enzyme, they'll go to where they should be on the histone?
That is what the cell's doing. We're still trying to learn the rules.
Right.
I mean, how smart is an enzyme? It has to know where to go, what genes to activate. So that's a good problem. How do you target these enzymes? But I think your first question is therapeutically. What are we going to want to do?
Yeah.
And I'll just say that if you had a patient like Jennifer Krantz, the job for us on paper would be then the histone mutation inhibits the writer. So on paper, we need to inhibit that inhibition. And so we've now at least found in the laboratory that the very modifications that Alfrey put on the map, if you put those into an oncohistone, it seems to make the inhibition of the writer much, 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 much less. So that's a therapeutic now avenue for us. And at least in the mice, that appears to be helping mice not get this brain cancer. So now it's going to be a question of how do we deliver that to a human being, obviously much later in time. Yeah. How will this pass the blood-brain barrier? How will this get into ultimately a therapeutically useful, situation in a human?
How about
but it's promising.
Yeah, I hate questions about time. How long will it be? It's always 5 years.
Well, the discovery, the discovery of the histone mutations was at the close of 2012. Now we're sort of sailing, through towards the end, I think, roughly of 2015. That's remarkable understanding of something that the DIPG physicians. I've had the pleasure to go to several NCI-based workshops where the job was you put the best people of your type in a room with the best physicians that treat this DIPG category. And frankly, it's just brainstorm, and it's been the most impressive, I think, educational and inspirational experience I've ever had. Because the physicians get up and say, we've got nothing for these kids. Now we've got a molecular pathway. Now let's work together. So I just came back at the end of August from St. Jude. It's my first time there. What a place. And again, very inspirational to me because just in the lobby, just waiting for my cab, you can see how sick these young children are. And, again, the mere thought, that sounds sappy and I'm sorry, but I think then, I say to myself, there's a percentage of them that may have maybe not the oncohistone mutation, but they've got a mutation in an epigenetic regulator. This marking system is catching really the scientific community by storm. And again, there's some promise because the DNA now hasn't been sort of irreversibly damaged. So we might be able to sort of reverse some of these mistakes.
And you seem to know now with this first drug how to successfully introduce an enzyme that will do its job. So you've got a process, it sounds like.
I think the cancer situations, primarily the blood cancers, were phenomenally effective epigenetic therapy targets because you can work with peripheral blood easily and so on and so forth. The brain cancers are going to be a challenge, I think, for sure. And so the tragedy of these super young children, I don't know, you asked for a time. I hope and honestly pray that we can, help folks like Jennifer Krantz, in the next 5 years so that there's a good treatment option. Nothing would make me happier.
How about other diseases that really take an enormous toll, and as time goes on, as people live longer, will take an even greater toll, like Parkinson's, Alzheimer's, obesity? Are these,
Absolutely.
It sounds like obesity you've already covered.
Well, I think, again, it's in its infancy with respect to sort of really crisp molecular insights. But, we'll take the neurological. I mean, I sit there and worry about things like that. And I'll just say that every time, not just our laboratory, but others goes and purifies a complex. That's what we do, probably best. We find, proteins don't run around just by themselves often. They exist in multi-protein complexes. And there's a protein in that complex that has a clear neurological syndrome. So, and frankly, probably the best accepted, first ever textbook epigenetic disease that was listed as an epigenetic disease was, I think it's contentious a little bit whether it's a true autism spectral disorder, but Rett syndrome. Many of you know that Rett syndrome is sort of in the sort of autism spectrum, and Huda Zogby and her colleagues at Baylor College of Medicine, they found a genetic mutation in a protein that binds this, to show you how fair I am, not a modification on the histones, but a modification on the DNA. So that protein binds a particular chemical group. That's the mutation that is in Rett syndrome, in essentially all the Rett syndrome children. But that protein that docks on the modification on DNA recruits all of these epigenetic writers and erasers. So suddenly, I think you might say, but that sounds like a genetic disease. Absolutely, the mutation in Rett syndrome is in a gene, but it's now causing a misregulation in the brain in genes that are either misactivated or mis-silenced.
This is such a complicated process with so many moving parts. Does advanced computation help? Computer
Absolutely.
computation?
Absolutely. I mean, we're not a bioinformatics lab, but I think we and all the other scientists here at Rockefeller and worldwide understand the bioinformatic and mathematical treatment of large data sets, will help genotyping individuals. And of course, then, I'm not a physician, but I think even in the brain tumor cases, the pathology of the tumors in the brain all look sort of in a microscope the same. But now they dig into the bioinformatics space and they mine and they mine and they mine and they find out, indeed, there's subtypes of these brain tumors. Even within the Jennifer Krantz DIPG, the anterior pons has different epigenetic marks than the posterior pons. So that really is going to be a bioinformatics.
Anterior pons?
The brain stem,
Oh.
if you just sort of cut it in half and took the anterior, posterior, there's different subtypes of cells and genomes, and importantly, the bioinformatics folks have found sort of, if you will, differences in the marking, the chemical marks on the histones and the DNA in the two different halves.
Seems like every time you open a door, there are 100 more doors. And
Absolutely. It's the way science is.
And that's why you really have to be careful how you count all this up, I guess.
Well, for, not that it's, the hour's late and you're saying, I get this guy off the stage. But in the human genome, since it has been sequenced, roughly 200 proteins fall in the general category of being epigenetic regulators. So some of the pathway I talked about, roughly 200 proteins or thereabouts, clearly are in that sort of general category, and they all have sort of cancer-causing mutations. So I think at least for cancer, this one is coming under close scrutiny for drug development, therapeutics, and this is a very exciting time for us.
Why don't we open it up to a conversation with all the other folks here?
Thank you so much. That was really interesting. I was wondering, since you do have information about genetically modified food and grain that we all are, consuming, and that affects the epigenetics of our particular generation, has anyone actually tried to do a connection or some kind of study in how certain populations that are ingesting genetically modified, particularly type of food, is sort of turning on and off these particular markers?
Well, it's a great question. Can everybody hear the question? Genetically modified foods. Has anybody done really a careful, systematic study of how that might or might not be? I'm not aware of any, but I'm sure that's my ignorance. I think, again, people really almost dismissed epigenetics as kind of a peculiar phenomenon. How could it really be that it isn't just all DNA? And so, I'm positive these studies are going on. I mean, there's hundreds of papers now coming into the literature that are doing just the kind of analyses that you're talking about.
You're both, I just want to make sure I understand, you're both talking about epigenetic changes in plants.
Well, this would be, possible that are, deleterious to us, that are maybe coming from taking in genetically modified. I can't give you a specific example or study, but I think that's a good question, and I'll bet money it's being looked at.
How does the pie that you eat now, long after you conceived your grandchild or your child, carry into that child?
Thank God I didn't have to conceive my own grandchild. But you're right. I mean, I guess now I can eat all the pie I want and just hurt myself. Yeah. But David brings up this really good point that it's something that we'll probably all be encouraged to think about in the future, that you're not only hurting yourself if you're still young enough to have offspring, that you have to think about.
I think I'm safe too. I can eat a lot of pie.
Yeah.
But so what we didn't really touch upon is the molecular mechanism of how it would be transmitted. That's going to immediately reduce itself to this marking system. How is it transmitted, during to the next generation? And that's going to become a problem of sperm and egg. And it looks like, in general, some of these modifications, these marks, hang on. They hang on to the sperm. Many of them are cleared. It's like nature has decided, better to start over. Let's erase the blackboard. But now what's intriguing as methods have gotten more and more sensitive, transmitted through the sperm or transmitted through the egg, there are modifications, these chemical marks that hang on. And now I think people are beginning to wonder, is that setting up a situation for then the next immediate generation? And then, of course, could that play out again and yet the, children, grandchildren, and so on? And I think I can explain this quickly, but I came back from a meeting just last week in Paris. Here's the experiment. Fruit flies have red eyes, easy to spot. That's a regular wild type or normal fly. But if you epigenetically silence the gene that makes it red, it'll become a white eye. That's easy. So how many people think that if you crossed a red eye, white-eyed fly, you'll get what kind of eyes?
Pink eye.
Yeah, see? And that's the Mendel, I grew up with that. That's the kind of situation. But this laboratory reported that once they jerry-rigged the epigenetic writers to be truthful, they could get a red-eyed fly that even if it crossed to a white fly, the children were all red. Next generation, all red. Next generation, all red. And then they kind of said, well, there's writers, and they're antagonized by erasers, so let's flip it all around. And then they flipped it all around, and no matter what the cross was, white-eyed flies crossed to red eyes, stayed white, stayed white, stayed white. And then the most amazing part, they repeated the experiments and just flipped the temperature. And suddenly the eyes went white, red. Unbelievable. And they flipped the diet, white, red. I said, whoa, this is spooky. So these are experiments. I think that's not how we all guessed pink eyes. And that isn't what this epigenetic sort of cross indicated. I think it's fascinating.
Based on the work and research that you've done and how our choices affect our health and that of future generations, are there certain things in our environment that we choose to ingest or we choose to expose ourselves to that you believe should not be allowed, that should provide implications for public policy?
Oh, wow.
Dr. Bloomberg?
I'm probably really not qualified to give any kind of advice, but I think now, you know, the one type of chemical flag, I kept all the chemistry out, but to be more specific, methyl groups, that's a simple chemistry, that's CH3 if you're a chemist. That modification occurs in a lot of dietary supplements. And the experiment where the mice, the mothers, the pregnant mothers, you know, gave rise to children that were different size and coat colors, very compelling. That diet was being spiked with methyl donors. So I don't know whether you wanted those mice children to be fatter or different coat colors, but it was dramatic effects. And so, you know, I think there's a case where the chemical modification is in common. Folate is the vitamin. And, you know, when I go for my physicals, I'll sometimes ask my primary care, should I be taking this? And it's like, I don't think so. You know, so who knows?
Could you find out in a dish if you had a population of histones of a certain kind and you added off-the-shelf vitamins to them? Could you see what it might be doing inside your body better than we now can with present research?
You could absolutely do that experiment with cells. And they could be human cells growing in a tissue culture environment. Absolutely, you can regulate the media, and you could then isolate the nuclei, isolate the histones, and with very precise instrumentation, we would know if these changes in sort of the media around which the cells, and then it would be more expensive and more challenging, but then you certainly could change this into a model organism where you fed the diet, again, extracted histones, but the question would be from where?
Yeah.
You know, which organ of the mouse or whatever are you going to look at? And of course, it gets very complicated quickly. But in the kinds of experiments where you could sort of then screen, sort of related to the question we just had, could you take human cells and start to screen them with anything you put into the media and then ask for phenotypic changes? Do you want the cell to grow better, faster? Or do you want it to sort of slow down? That might be kind of a anti-cancer therapy promise, that kind of thing. That could easily be done.
What about, you talked about different organs. What's the effect on stem cells and stem biology of epigenetics?
Very clear, not our laboratory's work, but you know, the famous 2012 Nobel Prizes awarded for stem cell biology, Yamanaka and Gurdon. Unquestionably, you know, if you think about my very first slide, we had genetics, we had the DNA, the gene, and then we had proteins that I called them activators. That's what Yamanaka and colleagues and Gurdon in different experiments found were absolutely essential to reprogram a cell quite effectively. But it isn't efficient, and it looks like what's in the way is this chromatin epigenetic layer. And there is one particular modification that is really a troublemaker. And you know, there's a couple of postdoctoral fellows in my lab. You might have noticed an acknowledgement for the Tri-Institutional Stem Cell Biology program that have now asked, you know, in reprogramming assays, what's the chemical modification that is the troublemaker? And again, too much detail, but it's a methyl mark on that histone. It's the one where the Oncohistone tracks the blood cancer. And when you sort of, again, poison that writer, reprogramming goes much better, much faster. So, you know, we're starting to think about that as a way to boost reprogramming efficiencies.
And by reprogramming, you mean?
To take a somatic cell and you now want it to be able to go backwards and then to give rise to other sort of,
to grow arteries or something like that.
or damaged nerves, or you name
Yeah.
it. The idea of regenerative medicine.
So until
The chromatin's in the way.
I'm sorry.
Chromatin's in the way. And so, that may be a barrier that needs to be overridden.
Until epigenetics got to this point, the idea of STEM biology hadn't reached this promising point, I would imagine.
Well, credit to the folks that won the Nobel Prizes. The mere fact that you could take a cocktail of we call them transcription factors, but to add those to somatic cells and get those cells to go backwards and differentiate into a wide variety of cell types was an unbelievable finding. So that, I think, boosts the argument for transcription factors as being critical, no doubt. Real genetics, no doubt. But the chromatin is in the way. So if you can find ways to sort of tip the balance, make it more efficient, that will only, I think, help true stem cell biologists.
That's so great. You've been fascinating tonight.